The next panel is assembled. The chairs, the co-moderators in this instance, are ready to go. And I'd like to welcome everyone to the second panel this morning. Our co-moderators are Joel Marcus and Lynne Zydowsky. Joel is a founder, CEO, president, everything, of Alexandria Real Estate, Alexandria Ventures, and he's co-founder and helps to run the Alexandria Summit, along with Lynne Zydowsky. Lynne has her own consulting firm, and she is involved as advisor to the ventures and the real estate. They're a group that have built this wonderful building in the complex for research laboratories, which is fabulous. Joel's background is to found and start these ventures and support young people as they start their businesses, and the whole idea is to use science to discover new therapies. Lynne is a scientist by background. She started in industry, in the pharmaceutical industry, and then migrated to her current role. Today, they are going to host this discussion of how you start a business based on an idea, on what you would do about therapy for unmet medical needs, which are critical, actually, to moving our industry forward. And unless we start these businesses, we will never get going. And the idea of starting business has become quite infectious at the university level, which is quite amazing to see, since I've been around it for many, many years, and see how many young people are starting college with the idea that they will start a business by the time they finish college. Quite amazing. Anyway, Joel, take over.
Thank you, Roy, and welcome everybody to the second panel at the pre-Galien. Want to thank the organizers and welcome to our house here. You're going to really enjoy this session. We've got some great people. Roy introduced Lynne, so I won't introduce her. Our bios are in the materials, but you're going to hear from three great people that Lynne will introduce a little more. Francois from the venture side, José Baselga from the cancer side, and Ryan Watt from the neurodegenerative side. And I think all of them are going to bring some pretty amazing insights. I kind of think today we are in a renaissance of venture capital. It's actually almost hard to believe. There are strong venture capital flows. There are somewhat fewer startups, although the numbers are actually staggering, but bigger startups in the life science end. We're seeing better financing and just much bigger companies coming forward. And I think there's also a favorable policy, you might say, in Washington compared to, the rallying cry of some of the kind of progressive sides about a single-payer system. It looks like if we can figure out how to make this system really work better, that the choice system is going to be the best system, and that's been good. I think the appointment of Scott Gottlieb to head the FDA, we had him here about 2 weeks ago, is nothing short of brilliant. He's amazing and is doing greatly innovative things at the FDA, and I think the NIH is doing well on funding. Venture remains very, very robust. I think, go back 1 second, today worldwide investment in R&D for life science today is approaching $200 billion, which is an amazing number. We're on track this year to, on the venture side, to potentially beat past years. So far, year to date, $61 billion in venture and over 6,000 deals across all sectors, which is kind of a staggering number. At the end of the third quarter on the life science venture capital side, we exceeded $14 billion, again, a pretty high watermark across over 1,000 deals, and the best year since 2015, which was $17 billion. Life science venture capital reached a quarterly high in this past quarter with almost $6 billion invested in a single quarter across 291 deals. Again, amazing. And as Jean-Francois will share with us, private biotech funding numbers for the third quarter have attracted over $3.6 billion in private flows, the largest quarter VC funding flow in the history. Much of the capital is also coming from traditional, non-traditional investors and crossovers, which is good. And philanthropy is adding something to the tune this year of $33 billion. Again, a pretty staggering number. So I want to turn to Jean-Francois. Well, let me ask Lynne to introduce, and then I'll go to Jean-Francois for the focus on early stage venture.
Thank you, Joel, and thank you, Roy. It's always an honor to be here at the Galien Forum. So to my left, I have Jean-Francois Formela, who's a partner at Atlas Ventures. His investments focus on new advances in biology, drug discovery technology, as well as novel therapeutics. He is the chair of the board of IFM Therapeutics, and he serves on boards of F-Star, Intelia, and many other companies. He began his career as a medical doctor and he received his degree as MD from the University School of Medicine in Paris and its MBA from Columbia. He serves as an advisor to Mass General Hospital. Next, we have Dr. José Baselga, the physician-in-chief at Memorial Sloan Kettering. He's also professor at medicine at Weill Cornell Medical College in New York City. His longstanding research interests are in the development of targeted agents for the treatment of breast cancer and in studying strategies to overcome methods of resistance. With over 457 peer-reviewed articles, he has conducted many pivotal laboratory and clinical studies that have led to the approval of many of the drugs that we're using in oncology today. And then on the far left, we have Dr. Ryan Watts, who is the founder and president and chief executive officer of Denali Therapeutics. Prior to that, he was the director of the Department of Neuroscience at Genentech, where he built the department and advanced multiple programs for neurological disease. He obtained his BS in biology from University of Utah and his PhD from Department of Biological Sciences in Stanford University. So thank you, speakers.
So, Jean-Francois, I've known you for several decades, right? Give us a sense of how you view the landscape today, how you view the areas of big interest to you guys at Atlas and other early-stage VCs in the areas of neurodegenerative diseases, cancer, and some of the rare diseases. And are we in a, every day you can turn on CNBC or any of the other financial news channels, and you see the indices ticking up literally every day, but then the commentators are saying, well, wait a minute, this can't go on forever. So are we in a boom, or are we in a bubble, or are we in both for early-stage VCs?
Well, if I knew that, I would be retired, right? If I could predict that, but probably a bit of both. My concern today. So on the one hand, of course, we're all very happy that the capital flow have been much better than for the past probably 20, 30 years. Venture capital in biotech since between 1995 all the way to, let's say, 2010, on average was $3 to $4 billion a year. That's what was going in biotech. You had a lot of variance. You quoted those numbers of 61 billion. Most of that is still tech today. Yep. And so that people understand the variance of the capital flow, in 2000, for example, biotech venture capital was still 3 to 4 billion. So, and that was steady, nothing was bad, but it was 3% of all venture capital flow, because tech flow in 2000 was 100 billion in venture capital. So going back to George's point, which was very inspiring, about availability of capital, it's still ironic that even with those numbers, there is a lot more money going to do, I wouldn't say pet.com or, silly things like that, but a lot of things that goes in tech and software and technology services, and there is still less capital going into healthcare and biotech venture capital, which is a very interesting way of looking at capital supply in our system, certainly in the US and in Western Europe. That being said, with the flow we're having today, which is much more comfortable than what we've had for the past 2 decades, you can see a flurry of investment. And so there is a renaissance. And the renaissance, the question would be, why is there more capital supply today? And why is it more steady? And we've seen an increase in capital supply recently in 2013. And we've seen a certain stability in the capital market. And in my opinion, the reason is that there are a lot of specialist funds that now have a capital base that is stable enough so that you can actually sustain the IPO market and the capital market in life sciences, which is a very important phenomenon. Now, the reason of the influx of capital, there must be a reason. That's the supply of capital. But the demand for capital has increased tremendously because of the incredible, I would say, explosion of scientific advance and technical skill set. The fact of the matter is today that the modalities we have available to us to treat disease are very different from what we had available to us 15 years ago. And for me, I'm just going to give you a little nugget, we can talk about it more, but people should pay a lot of attention to gain of function. For 50 years, or since the beginning of this industry, we've been a loss of function modality. Small molecule antibodies essentially are mostly inhibitors. Even siRNA, which was a beautiful scientific discovery about 15 years ago, Tuchel, Zamor, and Bartel, wonderful discovery, you're still degrading the message, so it's a loss of function. Obviously, you can degrade an inhibitor, which leads you to being a gain of function, but fundamentally, those are loss of function modality. Today, you have an explosion in gain of function modality. Of course, with the pinnacle being gene editing, which eventually, with one injection, if you actually edit right and you edit enough of the cell, you will essentially cure the disease with one injection and restore function. So that explosion of modality and technical skill is, fortunately for now, matched by an increase in capital supply. My concern is what happened, whether it's for geopolitical reason or for regulatory reasons, that capital supply start to actually decrease and maybe dry up, then we'd be in very big trouble. And in some ways, George might be right to be worried. But I'm still optimistic. I think we're going to continue to see some stability in capital supply. So I'll leave it at that. We can, I probably talked too much already.
So maturation of the industry, maybe a comment or two on how you think about that and kind of the boom and bust cycles. They seem to be somewhat more smooth these days. Would you agree with that?
Yeah, totally. So that's related to, I was talking about specialist fund, the influx, or I should say the outflow of capital out of those funds has been less marked when you have bad news. It used to be that if you had two big regulatory failure, a lot of those funds were very small. There was a small group of people. They had a small group of investors, and you could see the fund inflow being very marked, much more cyclical. Remember, the windows were open, and then remember in the 90s, we could have 2-3 years without one IPO. Maybe you had one or two, but you had literally one or two. And then you had a window, everybody was going out, because you knew that's when you needed to raise money. It didn't matter why, or if you had anything, or any reason, or any message, that's when the money was available, you had to do it. Today, the capital flow are much more stable, because the pool of specialist investor has a much more stable base of capital. And the cycle now driven by the generalist. The generalists, by the way, right now are not massively investing in biotech. I mean, there is some money, but a lot of that money that you were mentioning is coming from specialist buy-sider who now are investing in private investment.
So the Cambridge Mafia, you guys at Atlas, Third Rock, Flagship, why are you putting so much money into much bigger startups than, say, the last decade or two?
So actually, we don't. We do more than we used to do, so we'll do Series A's of 30, 40 million. TRV came in the market with a very interesting strategy, which is they said, we're going to start with 50, no matter what, and then we're going to go raise another 100. I mean, I'm being a bit simplistic, but there was a deliberate component of the strategy to give a lot of capital to the company early. What people don't realize that our models are not that different is that the gestation period, A friend of mine was joking that TRV gestation period is like an elephant. So you have like, I think it's 22 months no matter what. And you know, when we do a seed, sometimes we're ready to do a Series A after 6 months. We don't really have that gestation period, which is almost fixed. And TRV strategy, which is very interesting, is to really build something big and also to go public, which of course means that they're more correlated than some other venture firm, but they've done an amazing job and they certainly hit the market right. So they've been incredibly successful and they've created wonderful companies. In our case, we're much more agnostic. We don't like to be too correlated with the public market, so we make some big investment and some smaller investment. In other words, we start with the science first. It doesn't matter what shape it has. It can be gene editing, where we raise 300 million in 18 months, because that's what you need to do. Or it can be a single target, something we like very much. I'll take an example. We looked at an SK2 channel inhibitor, so it's a cerebellar ion channel. It was one target and one disease, which is ataxia. In fact, it's not even the disease, it's a symptom of ataxia, which is a motor symptom, which is the most debilitating for those patients. We liked the target very much. It was a perfect correlation between the target dysfunction and the phenotype, and whether in genetic model or non-genetic model. So that was not really a company, it was a program, but we liked it so much that we funded it. And we're actually going to be in the clinic next year, and we're excited about it. Now the question is, do we bring some other program to build a company around it? It doesn't matter to us, because we're very agnostic to the business model. It doesn't have to be a big company that goes public. 2/3 of our exit are M&A. So that program, if it's valuable, will be bought. It's not 90% chance, it's 100% chance. And maybe I'm a bit over-optimistic, but if it works, it will be bought. So anyway, go ahead.
So one other question. In June, you guys raised a $350 million fund. Was it your easiest raise? How do you look back on that? And what's the life of the investment on this fund given today's market?
Yeah, so look, part of it might have been the climate. As an investor, you know that we've been fortunate in the past few funds, and that was the fastest, easiest raise we've ever done. So it was less than 3 months from launch to closing.
Pretty amazing. And how long will it take you to invest, given what you know about today's marketplace, do you think?
So that's a very important question, because when you are in a cycle like that, if you're in a good cycle and you've had good performance and you can raise a lot of money, so we were probably about 3.5 times oversubscribed. So we could have raised, I don't know, maybe 800 million. That's exactly what you do not want to do, because you cannot be changing a strategy overnight by doubling the amount of capital and assuming that you're just going to kind of do twice as much as what you did. Because there is a very subtle relationship between the amount of capital and the discipline, the strategy, and the quality of the investment you make. So you grow a little bit. We went from 280 in the past fund to 350. But because we're not focused on capital intensity, in fact, we're focused on lack of capital intensity. We like plays where you can get to a lot of value on less than 30 million, because if you do that, you only need $300 million to do 10x. If you do something where you invest a billion, and I'm actually very supportive of those strategies, so I don't mean to criticize my colleague here, but it's a different equation. You're relying on different parts of the capital market. And for some of our play, we do that. At Intelia, we're going to be relying exactly on the same model they do. In some of the other plays, we're relying on just very, what I call rifle M&A.
So Lynne? Thanks, John.
Thank you. So I'd like now to go to you, José. In addition to being the physician-in-chief of Memorial Sloan Kettering, you're also the head of the SAB and a member of the board of directors of GRAIL. Many of you probably have heard of GRAIL. It's one of the biggest new startups based in Silicon Valley, a big data company that sits at the interface between life science and technology. They bet on that the cure for most cancers wouldn't be a new drug, but tests that catch tumors at an early stage when they can be treated. And there's the statistic that roughly 2/3 of cancers caught early are curable, often with surgery, while those caught late are 80%, often fatal. this could represent the single biggest diagnostic opportunity that we've ever seen. So I'd like to turn to you, Jose, maybe talk a little bit about the platform and what this could mean in terms of impact for human health and healthcare overall.
And how much you raised.
We'll get that's the next question, Joel.
That's the next question.
That's my first question.
So I'll keep on talking about the first question to delay the second question. So the promise of GRAIL and what's it's so exciting is basically what has occurred with our capacity to sequence DNA from tumors and the availability of looking at the DNA or nucleic acid from tumors in the blood. So that's the concept of the liquid biopsy. We have the capacity today to sequence massively and at a cost that is coming down. And therefore, the promise of identifying DNA from the tumor in the blood at a very early stage is a reality. And we have now, and we and many others have data that support this. And the premise, therefore, is that if you were able to develop a test that would identify and would detect cancer earlier, we would have a shift from late diagnosis to early diagnosis, and that on its own would change survival in a very major way. So, for example, let's take, as an example, breast cancer. Today, if we were to diagnose our patients with breast cancer at stage 1, the cure rate will be, for everyone, north of 90%. That would be a huge shift. Even in tumors that are very tough to cure today, such as pancreas cancer, if we were to diagnose stage 1 pancreas cancer, cure rates are north of 60%. So, therefore, if we could develop a pan-cancer test that we could apply in the population as a whole, as a screening test that would perform with high sensitivity and, very importantly, high specificity, then that would be a true change in the way we approach cancer diagnosis. So that's one component to that. The second component is that these liquid biopsies are not only telling us that there is cancer, but it's also telling us the features of that tumor, is giving us opportunity to treat those tumors better, more targeted, because we will be able to untangle the genomic component of that tumor, and therefore be able to predict outcome and to identify as well the treatment. And then finally, in patients that we have that have a high chance of cure from cancer. Let's imagine a patient that goes through surgery and chemotherapy. The big question that they always have is, am I okay? Am I cured? Am I tumor-free? And today, we cannot answer that. And it's a big issue. And, for all of us who take care of patients, is perhaps the most highly quoted question that they have, am I okay? I think with these technologies, we'll be able to know that something is not okay or something is okay and intervene much earlier. So I think is a true breakthrough technology. The question, of course, is how do we apply it and how do we bring to the market something that will be useful for the whole population?
So to do this, you need to really scale.
Yeah.
And as Joel alluded to, you raised early this year, you've raised over $1 billion, an amazing financing that has hit records that we've never seen. Some of your investors are Illumina, Arch, J&J, BMS, Merck, Celgene, Google Ventures, Bill Gates, Jeff Bezos, and I'm sure there's many others that I'm not mentioning.
Alexandria.
But, truly remarkable. And maybe just talk a little bit about, the aspect of scale. So I saw this interesting statistic scientifically about GRAIL, that it said it collects 1,000 gigabytes of raw data from each volunteer's blood, and it's equal to 500 hours of movies. So let's talk about scale.
Yeah, so this is really an operation of a scale when it comes to data that we had never seen before. Just to give you an impression, for these trials, we are enrolling thousands and thousands of patients to the scale that had never been seen. People are talking about pharma studies in phase 3 studies, and a large pharma study may have a few thousand patients. We're talking about studies that will have hundreds of thousands of patients that we will follow prospectively. And therefore, imagine the magnitude of the sequencing effort. Well, first of all, the logistics of it. Then, secondly, the magnitude of the size of the data. How do you interpret this data? And then how do you execute on the findings? So this is a scale that is really, it makes you pause. But at the same time, if you don't apply scale, you cannot go incremental, because to the point that George was taking, I don't want to be back here in 2025 saying that the studies are still ongoing. So we can't do that. So the classical approach would be to take one step at a time, but that will take us so many years. I think that we can do the studies that are definite, that are giving us a good answer, and for that we need to enroll hundreds of thousands of patients, and that's what we are doing.
It's really remarkable. I'm going to just segue over to Juno, another company that shares a lot of the same investors, and you, the Fred Hutch, and Seattle's Children's Hospital were very involved in the launching and the founding of Juno. This is a company that was started in 2013, was able to go out, raised hundreds of millions of dollars, was able to go out and go public in December of 2014 in a record-breaking $265 million upsized IPO that was 30x oversubscribed. It now has a market cap of around $5 billion. It's focused in immuno-oncology, Karthi, which is in the news. There are over 300 trials right now going. That was a statistic I read in Karthi. And we've seen recently 2 drugs that have been approved with Novartis, and then Kite, which was recently acquired by Gilead. So maybe you could talk a little bit about the area and the promise of these drugs.
Right. So the Karthi Cells, the concept is incredibly elegant. And the science is quite strong. So basically the idea is to take the T lymphocytes from a patient, take them from the body, and then in the lab introduce a receptor that identifies a specific antigen that is present in a tumor cell. Those cells, these receptors, upon binding, activate a program that basically results in killing the cell to which these T cells are bound to. So then these cells are expanded and they are put back into the patient. So in a way, this perhaps is the first living cell therapy that is out there. And the concept is incredibly elegant, and the concept is one that we have seen great results. So the Novartis compound, and this was technology developed by Carl June at Penn, have shown dramatic responses in children with leukemia, with ALL, and huge complete remission rates. Then there are a number of data in lymphoma, also showing very impressive remissions in patients refractory to disease to therapy that had received 2 lines of therapy, and they had progressed. And then even more recently, we have quite amazing data in multiple myeloma. The data in myeloma, it's even better than anything we've seen with lymphoma. And we believe that this is just the beginning. Now, this is a challenging disease, a challenging therapy, I mean. You've seen the price tag that is coming along. We are crunching numbers at every single hospital, and I don't know yet whether we'll be able to deliver this in a way that is efficient, and also it's toxic. I mean, these lymphocytes can get off hand, and we've seen some toxic deaths with some of the compounds. So it's going to be a challenge, but the concept is incredibly powerful. And also, it can be used in a much broader sense that is being used now. So until now, it's been used mostly to have the T cells kill the tumors. But you could also envision that this could be used as a delivery system. So you could use these lymphocytes, and you could load them up with anything you want. And they are like FedEx, right? Or they are like DHL. You just go in and you deliver your load or your message. You say hello to the tumor. I'm coming. So I think it's just the beginning, and it's just it's a beautiful concept, I think, and very appealing.
So I'd like now to move to Ryan Watts. So yet one more example of kind of this large investing in a very hard area of unmet need, and that's the area of neurodegeneration. Denali Therapeutics was founded in May of 2015 by Ryan, who, as I said, was former head of neuroscience at Genentech, his second co-founder, who was also at Genentech, Alice Schoof, and Mark Tessier-Levine, who's the president of Stanford. You work on all the hard neurodegenerative diseases, ALS, Alzheimer's disease, Parkinson's. We know that the patients who have these diseases represent a staggering cost to society. And I thought I just want to focus for a minute on Alzheimer's and just read, some statistics. It's the 6th leading cause of death in the United States. Alzheimer's has no survivors. 1 in 10 people over the age of 65 have Alzheimer's dementia. That represents today 5.5 million Americans of all ages. And that number is projected to increase to 7.1 over the age of 65, and by 2025 and by 2050 expected to double. The direct costs to American society for caring with those who have Alzheimer's is estimated in 2017 to be almost $260 billion. And if you do the math, that statistic comes out to more than 20 million per hour. We have 1 in 5 Medicare, Medicaid dollars going to Alzheimer's, but with the 71 million baby boomers that are aging into this high risk for the disease, that could increase to 1 in 3 billion. In addition, 15 million Americans provide unpaid care for people with AD. The economic value of this care is estimated to be $230 billion in 2016. So it's a growing epidemic. It's really starting to challenge heart disease and cancer as the leading cause of death. And if left unchecked, it could bankrupt healthcare systems everywhere. We've seen neuroscience venture funding increase in the past 5 years by over 40%. And maybe just talk a little bit, Ryan, why this has been such a difficult area and why now we're seeing, this renewed or this interest in putting a lot of dollars into this area.
Yeah. Thank you for the question, and thank you for the invitation to be here and speak about Alzheimer's and neurodegenerative diseases. Usually when Lynne invites me, she starts with the failure rate. I don't know if you remember, this is maybe the 3rd or 4th time I've been on this, and we talk about the fact that we've failed in developing medicines in this really difficult area. But before I do that, I just out of curiosity, and you don't have to raise your hand, but my guess is that 1 in 2 of you in this room has a grandparent or a parent who has dementia. Probably 1 in 10 or maybe 1 in 20 that has Parkinson's disease. And for those unfortunate, those of us that are unfortunate, you may know someone with ALS. There are no disease-modifying therapies for these medicines. We've been talking a lot about investment. I think what's happened is the vision that if we can solve these diseases, it'll be incredibly transformative to the medical system. Just speaking personally, having seen Alzheimer's in my family's life, and as I'm sure many of you, it's devastating. And I think we had a really great opportunity here 2 weeks ago to hear from Greg O'Brien, who was diagnosed with Alzheimer's disease about 8 years ago, and he shared his story. I was actually very nervous for that lunch, actually more than any presentation I've ever been involved with, because I met with Greg before the lunch meeting, and Greg and I spent about an hour together, and he repeated many of the questions. He actually got lost, going to the bathroom. We didn't know where he was for about 15 minutes, and he made a joke about it. I loved his spirit, his sort of unconquerable spirit. But I was nervous that we'd get up there and Greg we don't know where Greg would go. Luckily, he read carefully from his notes, but what was amazing to me was how his disease has impacted his entire family. And his 28, 29-year-old son basically is now a full-time caregiver for Greg, who's in his is in his mid-60s. I've seen it, in my family. I'm sure as many of you have as well, where one of your parents who is otherwise healthy has to retire or has to give up something to basically care for their spouse. And I think when you look at the investors that have gone into, let's say, GRAIL, I know this is where we'll go, and Juno and Denali, the idea is we want to solve big problems, and this is a big problem. So the question is, why now? If you have a 99% failure rate, why do we think we can solve Alzheimer's today, and we're unable to do that, 10 years ago or 20 years ago? And let me tell you what's really changed in this field that makes it, for me, probably one of the most exciting fields in terms of the science. Some of which we contributed to while at Genentech and certainly while at Denali. In 1991, the first gene was discovered that was associated with Alzheimer's disease, is known as amyloid precursor protein, or APP. There are about 2 or 3 other genetic associations that came over the next 2 or 3 years. For example, in 1993, I'm sure many of you have heard of ApoE4. So actually sitting here in this audience, 25% of us will carry the ApoE4 allele. I'm an ApoE4 carrier. It's very common. If I carry 1 copy of ApoE4, my increased risk of developing Alzheimer's disease is 3.5-fold. If I carry 2 copies, it's 14-fold. Maybe that's why my mother has Alzheimer's disease. But actually, if you look at the statistics in the Alzheimer's population, 70% of Alzheimer's patients have ApoE4. It's 60 to 70%, depending on the clinical trial. And in the general population, it's 25%. That was some of the early, what we call degeno genes when mutated that cause or a major risk factor for Alzheimer's disease, very, very similar to the oncogenes in cancer. As we started to understand the oncogenes, we started to develop therapies that were very targeted. And what's happened, thanks to Illumina, which of course is one of the founders of GRAIL, since 2008 and today, we've gone from 2 to 4 degeno genes in Alzheimer's to 30 or 40. And what's been really, really interesting is that we used to believe that the inflammatory response in Alzheimer's disease was really just a consequence. Neurons die, microglia, which are the resident immune cells, become inflamed, and basically the disease is worsened. The genetics has told us that greater than 50% of the genetic associations are expressed exclusively in those resident immune cells. This now points to a potential for a direct causative role with a defect in the innate immune system. We can call it, neuroimmunology, not unlike cancer immunology. So we have several programs. We in fact have 2 programs focused on the neuroimmunology axis and going directly after some degeno genes, these genes that are mutated, that are risk factors in Alzheimer's disease. There's been one other major barrier, and I use that word intentionally, to drug development in neurological diseases, but also in neurodegenerative diseases in particular, and that is the blood-brain barrier, which is a challenge for us to get medicines across the blood-brain barrier. We did this analysis of sort of the failure rate. We looked at all the drugs that had failed in Alzheimer's disease, and we asked some simple questions. And this is in phase 3, why was a specific dose selected? What was the evidence around that dose, that drug engaged its target in the brain? What was striking to us, that at least in the public databases, these are now trials that have failed, and drugs that you can make and test, that basically greater than 80% of those drugs had no evidence, at least reported, of target engagement in the brain. And so those are non-experiments, right? Those are basically experiments, and that's completely changed. In fact, you've seen it. I think one of the most, one of the more recent exciting pieces of data was from Biogen on their aducanumab approach, where they're now seeing a pharmacodynamic response. They see plaque reduction in a dose-dependent fashion. So at least we can say, hey, we're going to go after, we're going to take a dose forward that we know is going to engage the target. So we really think that there are two things that have come together in this field. One is our understanding of the biology, and we can talk about Parkinson's if anyone's interested, as a result of the human genetics. And the second is we've actually improved our understanding of the blood-brain barrier and how to engineer molecules to cross the blood-brain barrier. And this is something at Denali that we're heavily focused on, is basically building platforms to get antibodies, to get enzymes, to get large molecules into the brain, and then when we develop small molecules, to ensure that they have the right chemical properties and the biomarkers to essentially ensure that you have targeting engagement in brain. So I think these advances in technology and understanding of disease make this time unique relative to the last 20 years of development in this space.
So Ryan, recruiting patients into your clinical trials, tell us a little bit about the challenges with that.
I mean, there are many challenges. I'll tell you one that's different. Let me just give you an interesting statistic. There are 6,000 plus ongoing phase 2 and phase 3 trials in cancer. 6,000, right? There are only 300 if you look at Alzheimer's, Parkinson's, and ALS combined. Okay, so you can see that, obviously our explosion in the understanding of cancer has resulted in, I think rightly so, many molecules being tested, advanced in the clinic. So there is a sense of, I won't say that there isn't as much competition in terms of recruiting for patients, but I think the biggest challenge we've had in some of the failures in the past is recruiting the right patients, the right phenotype. And I've been part of a phase 2 clinical trial where we did not pre-select, for example, for amyloid. If you have plaque in the brain, we discovered after the fact that 30 to 40% of the individuals enrolled in the trial don't even have the drug that don't even have the target that the drug is going after. Right. So I think what you have to do is you have to have these biomarkers, imaging biomarkers, blood-based biomarkers, pulse, cerebrospinal fluid biomarkers, and so that's one key is making sure that the patient has the target. The other major challenge is the timing of intervention, and so we participated in the first ever Alzheimer's prevention initiative. Some of you may have read about it either in the New York Times. There was a Nova special, "Can Alzheimer's Be Stopped?" That talked about this basically mutation in a gene that causes Alzheimer's disease at age 40, and it's found. It's in Antioquia, Colombia. So there's about 5,000 kindred, and the idea is that we can treat those patients. The way that we're running that clinical trial, and when I say we, I mean Genentech. So obviously I've moved on. Is enrolling at age 30. These individuals are otherwise healthy, and we're basically looking for delaying the disease. And so I think finding the right patient, either a genetic population, having a biomarker to make sure the target is there, and thinking about time of intervention. So those things make this area very challenging in terms of patient identification, recruitment, and making sure the trials are designed appropriately. I won't even go to the clinical endpoints. That's a whole nother piece, right?
So Jean-Francois, as you listen to cancer, neurodegenerative diseases, how do you guys think about, obviously, huge opportunities? How do you think about the end game, which is how patients get these and how they get paid for?
Yeah, so I have to say pricing is a concern, but it's very hard to incorporate pricing into an early stage venture capital strategy. In some ways, the analogy would be when Mark Fishman came to Novartis, Dan Sella hired him. Great idea, which is probably one of the reasons why Boston exploded, within the following 12 years. Mark was famous for not caring at all about what the commercial guy was telling him at Novartis, right? Which, in fact, has been source of fun, interesting stories in tension. And he's been proven, I would say, in large part, right. There has been a number of disease. Jean-Paul Clausel, the founder of Actelion, right? So we did the Series A with Jean-Paul many years ago. And we just had an interview with him at our CEO retreat, and he's a commercial guy at Roche. We're telling him that PAH would never be more than $200 million peak sales worldwide, right? And they stuck to it, and that's one of the reasons why he got out and he started Actelion. So that's, of course, a slightly different issue than the issue of pricing. My point is that it's the same variable. It's very hard to integrate, the end economic value when you're actually investing into a target and you're actually investing in chemistry. And so I think at the end of the day, Mark and others now in the industry were probably right, which is science first. What is the unmet need? And what is the tractability of the biology and the target? So that's our primary criteria. Once you have a truly novel biological target, and obviously genetics plays a very big role now. I think the industry used to be a little bit less concerned about genetics and validation. In fact, it's very interesting. The CF program at Vertex was originally a phenotypic screen at Aurora, which was an afterthought. And there was no SAR, and they didn't fully understand the mechanism. It turns out that it's an amazingly interesting mechanism because essentially those small molecule are protein correctors. They rectify or they correct the protein to enable it to function to a certain extent. But the fact of the matter is that for us, it's all about science. Initially, novelty, what would be the potential impact, independently of the payers. What we believe is that if you really bring value, you're going to be paid. And the question is, maybe it's not going to be as much as you would have hoped, but I think it's going to be enough. I mean, for me, the ultimate example is Gilead and Solvati. I think that the industry and the policymaker, and we, all of us have been, of course, talking to people in Washington. I think some of them are not entirely, dumb or dishonest. Some are actually trying to do the right thing. And the fact of the matter is that, Gilead got screwed. That price is an incredible amount of value. There will be tens of billions of dollars in savings that will accrue to the healthcare system for the rest of time. And Gilead would only get an infinitesimal portion of that. So that's a long answer to your question. We don't worry too much about pricing. Science first, tractability, unmet need.
Yeah, so I'll just make one comment on the Gilead matter. You have to always think about a cure and an insurance company paying for a cure. If they capture you for 1 year under a policy, their thought is, well, better we just maintain you and take care of you for the year because we may not have you next year. So a cure may not be affordable to today's current insurance company structure. That's one of the problems that I think we face and we've got to solve. But let me move to José for a second. And I'm not asking as an investor, but I'm asking in this forum, how do you think about the business model of GRAIL?
Well, I think
because it's so novel.
Yeah, so I think it's still very unclear because it's all going to be based on the data that we have and the findings, and then also it's going to be highly dependent on the payers and so on. But the model is going to be very different from what we've seen ever before because. By sequencing the tumor, by identifying tumor early, you have a lot of information that you can provide to help taking care of that particular patient, right? So I could see a situation in which we could work very closely with insurance companies and that we would add value in a way that is very different from just developing a drug. This could be a global health company in that we are able to provide information not only on the outcome, but also on how to treat that particular case and also how to follow the response to different therapies. So it's going to be incredibly complex, but look at what GRAIL will do. It will touch at the diagnosis level, at the management level of the disease, and also certification of risk factors and outcomes, and then also at monitoring if any given therapy is working. Let's look at the later part for a second. Today, we are using very expensive drugs, and we use them, for example, chemotherapy or some of the monoclonal antibodies, some of the immune therapies, and we have to treat those patients for months before we even know if it's working or it's not working. So classically, we start a new treatment, very expensive, with substantial side effects, and it takes us 3 to 4 months until we know that that particular therapy is working or not working. With nucleic acids in the blood, we can today, and we have a number of examples, be able to identify if a therapy is working within 10 days, within 2 weeks. That could change tremendously the way you basically are delivering care and the way you are using certain drugs. So I don't know the way this will look like, but this is going to be very different from what we have seen, and it's going to be very interesting to see how this plays out.
So one other question for Ryan. Tell us about your vision for ALS, the Lou Gehrig's disease. All of us have had friends or families who've seen the debilitating disease that has been so intractable. How are you guys strategically thinking about that?
Yeah, without going into great detail of all the the programs we're going after, there's obviously the same advances in genetics. I think a great example is there's discovery of C9orf, but related to ALS, I had a very interesting experience. We've just a little bit of our history. We were founded 2.5 years ago, and there were 2 of us. We have 125 people at Denali today. 60 of them are in discovery, so they're scientists working on the biology and inventing drugs, and 30 are in development as we're in the clinic now, working on Parkinson's and ALS. We have a molecule that targets, again, that neuroimmunology axis in ALS as the disease spreads. And we actually, what really striking, I have a very close friend who was diagnosed with ALS 8 years ago. He's 35 years old. He was working for Microsoft at the time and was training for a half Ironman, and basically started to develop weakness in the shoulder. Actually, anytime I feel like sore or weak, I get nervous because when you work on these diseases, you're just constantly afraid of them. And basically, went on to develop further weakness in the right arm, and then over about a 2-year period. It took a very long time to diagnose, and I think about the power, in fact, of genetics, but also in diagnosis for these diseases. Our challenge often with ALS is that it's misdiagnosed for a long period of time, and by the time you have pretty severe degeneration, that's when you're looking at therapeutic intervention. But anyway, Seth, my friend with ALS, came to Denali. And I think if you can imagine for a moment, the scientist who invented our drug, which will actually file to enter the clinic in January, he came and presented for an hour and told us about his diagnosis and how it impacted his family of 4 children. It was one of the most powerful experiences. But what we realized is, obviously the great unmet need. There's one drug approved in ALS, riluzole. It extends life by about 3 months. And the mechanism is not well understood. But just like Alzheimer's since 2008 and today, and in Parkinson's 2008 and today, really the last 8 or 9 years, we now have, again, 30 or 40 genetic associations in ALS that point to, for example, disruption of the lysosomal system, RNA, DNA homeostasis. So these RNA binding proteins appear to be mutated and cause a defect in handling RNA, and that's a theme that we're seeing across many of the genetic insights. So we're going directly after that, and then we need some help in being able to diagnose, a little bit earlier, so we can intervene if you're going to go after these the causative targets.
So why don't we open it? We've got 15 minutes left. Yep. Oh. Do we have a mic up here at all?
If not, I could speak loudly.
Oh, I can just repeat it.
Okay,
So.
very good. So really a question for maybe José and Rob.
And just introduce yourself for a second.
Hi, I'm Bill Chin. I'm at Pharma. Great panel. For José and Ryan. So for José, and both of you, it's really about how we think of diseases how we're going to treat them in the future, and we talk a bit about maybe it's more likely we'll be able to prevent disease or stunt the progress of disease early rather than maybe treating it. So that's what we think now. So for José, I'm very curious. Assuming Moore's law will play its role out and you can be able to screen and do tests, maybe whole genome, whole exome screening for everybody at age 20 or whatever. What is your concern that maybe there will be a number of false positives, especially given the advances in immuno-oncology now? One could guess that, in fact, we all are developing tumor cells on a regular basis, but our immune system actually takes care of it. What if you detect that change, but then ultimately you really don't know? And maybe with Ryan, really sort of the issue of biomarkers, you said the major challenge is the blood-brain barrier, but I would argue the major challenge is developing biomarkers that will allow you. It's almost chicken and the egg. What comes first? But what about using the blood-brain barrier and understand what comes out to use as biomarkers? So those two questions.
Yeah, so you were raising one of the biggest fears that we have, and actually in my opening statement I referred to the need to develop diagnostic tests that would be highly sensitive, but they have to be specific. And look at what's happening today with mammography that is full of false positives. We cannot have tests done in people, healthy otherwise, and tell them, you have mutations. You might have cancer. I'm not sure. We'll see. That is not doable, and that would be totally wrong. So there are going to be two approaches to this. One is that by looking not just at a few panel of genes, but rather by looking at a broad number of genes, like whole genome sequencing or whole exome sequencing, we will have algorithms that will be quite precise in telling us that what we are seeing is beyond the doubt cancer. And also, we might have to introduce a temporal follow-up. So we might need to have, as what has been done by Dennis Law in Hong Kong with the AVV, temporal confirmation. So maybe we will be talking about a concept that the virologists have done very well, which is viral load. So maybe we need to introduce a concept of tumor load and have one assay on month number 1. And then if we are not sure, we just repeat the assay 3 months later and see what's happening. And yet, we're going to have information that we don't know how to process. So the classical example is what's happening today already with a concept that is called a clonal hematopoiesis. So when we sequence today the blood of healthy people in an age-dependent fashion, at age 70 and older, about 20% of the population have mutations in the white blood cells that are identical, totally identical, to the mutations that we see in leukemia. You cannot distinguish them. And we know that this population have a higher risk of developing leukemia, but we don't know at which rate, and we don't know when do we need to start treating, because maybe that's an opportunity to start treating. So we are going to be opening next year at our place at Sloan Kettering for the first time a clonal hematopoiesis clinic, in which we are going to be following this population and we're going to learn. So yes, you are right on, and we cannot have the same problem that we have with mammography and with false positives, because that would be unbearable.
That's a great question. So your question was around biomarkers as being as big a barrier as the blood-brain barrier. And we really identified, I think, 4 areas. And I'm sure after, if any of you want to come up and tell me like 5 or 6 or 7 other, areas that are limitations to success in our field, I'd be happy to hear it. The first is a limitation on the targets, and there it's really genetic pathway potential. We have now many targets. The second is the blood-brain barrier, and now the focus on engineering brain delivery. And the third is biomarkers, without any question, a limitation of biomarkers. And for us, it's all about biomarker-driven development. And I think about it in 3 ways: target engagement, pathway engagement, and disease biomarkers, right? And so it depends on what type of biomarker we're talking about. But a lot of times we fail at the first, which is the drug engaging the target. So we have to get that one right. The pathways now, like glial biology, lysosomal function, working on pathway biomarkers for disease, and then finally disease biomarkers like imaging, and there we're seeing major advances in imaging, but certainly that has been a barrier to our success. By the way, the fourth one is just being rigorous around translational medicine. It's basically using the principles of dose selection, target engagement, patient identification, right? So if you look closely at this field, the biggest advance has really been in imaging biomarkers and fluid-based biomarkers. And I think actually one of the most exciting things, talking about, you asked, using the blood-brain barrier as a biomarker, is when neurons degenerate, they appear to release what's called neurofilament. It's a specific, and it happens to have pretty good kinetics. So neurofilament that's produced in the brain can actually be, gets through, not the barrier, it's actually cleared through ISF, CSF bulk flow through the arachnoid villi, and then it has some level of kinetics, so it doesn't get degraded immediately, and you can actually measure degeneration happening in the brain by looking at blood and neurofilament levels, right? Now, this is certainly the case in ALS. It's not in every neurodegenerative disease, but definitely the case in ALS and FTD, and so that's really exciting to think about that as a way of potentially measuring degeneration, the levels of degeneration.
Thanks, Bill. Other questions from the audience? Yes, sir.
My name is Michael Harbour from Merck. And at the outset, you talked about approaches in venture capital and investments, and you mentioned the $300 million mark, Dr. Formela, regarding what's an ideal amount of money for you to develop a portfolio of companies to invest in. But then we heard about GRAIL from Dr. Baselga with a $1 billion investment for just one company. And I'm wondering if, and you intimated that there was some difference in approach there. And I'm wondering if you could talk about that a little bit more, if that's more of a testament to the end results of what we're expecting from GRAIL, or if there's something different here about this particular investment per se that made this investment so large, and or is it going to be too big to fail, or can it fail at this point? So I'm just like a little bit more insight into that.
So I would say, I don't to be overly simplistic, but the difference is arithmetic. $300 million is not numbers that we have in a plan. It was an example, right? I mean, if you invest 30 million, which still is probably an average size for a lot of Series A, it would tell you that you need 300 million to do 10x. But 10x is not. A lot of people would kill to have 10x. I mean, a lot of venture capitalists are happy with 4x. And when we make an investment, again, we're not looking at what is the required multiple. I mean, of course, there is a minimum required multiple, but it's science first, tractability, try to bring value, and you know that if you succeed and you have a program that is differentiated, hopefully novel, and has a very good chance to provide a signal in phase 2B or even in 1B, there will be a buyer, and that comes from you guys, right? I mean, the pipelines, there is an incredible need for pipeline. We know that a large fraction of innovation in this industry comes from biotech companies that are then being acquired or partners with pharma. So let me talk about the two models. As I said, in our case, we're agnostic to the business model. So if it's a single target in a disease that might be an orphan disease, but we believe there is a great unmet need, great science, and it's tractable, we believe that we'll make money by virtue of pure arithmetic, which is we don't need to spend as much, and the exit value will be proportional to value, and you'll probably have a good multiple. I think José and his team are trying to solve a much bigger problem, and the capital requirement to solve it is just different. So you have to adjust the business model. It would be sad if there was no one in the venture community, God bless Bob and a few other people out there on the West Coast, that were not willing to gather a very strong syndicate of unconventional investors.
Right.
It'd be very hard to do that with venture capitalists.
Right.
But Bob has been very good at putting together corporation, pension fund, venture capitalists, to raise the money necessary to solve the problem. And I think I would assume, and José can comment for himself, they have the same expectation we do, which is if you solve the problem, the value will be proportional to the size of the problem you solve. So here you are. I think it's a wonderful thing that you can go attack problems like that.
Yeah, so I think GRAIL is a little different. It's not your classical discovery company, right? If you were to discover a new therapy and you were going to invest upfront $1 billion, I think that would be complicated, right? How to go about that. But you need to see GRAIL as the science is here, right? The science is here. The technology is here. And it's about execution. So what we are facing is an execution issue that we need to deliver on. And to do that, we need to do studies in hundreds of thousands of patients, and there is no other way around. So I think that when you have a very clear roadmap of what you want to achieve, and you have everything in place, that's the kind of capital you need to be able to deliver on this in a timely fashion. And I think that's the different model, right? I mean, we know exactly what needs to be done, and we need the funds to be able to deliver on it.
And I think the concept in GRAIL was to have enough of a war chest to do what José said, probably for a 3 or 4 year period when you can focus on just execution as opposed to constantly hitting interim milestones and trying to raise incremental money. So we have time for maybe one more question. Yes, sir.
Good morning. My name is Demetrius Angelis with Pharmaceutical Patent Attorneys, and this is maybe more for Jean, Francois, and Ryan. We have a lot of clients who are at the early stage of their development where they've got strong patents and lots of good preliminary data, but when they go to get venture financing, they're told we need phase 2, phase 3 data. And of course, to get that data, they need millions of dollars. So what do you say to folks that are in that sort of catch-22 where they don't have these access to this large amount of money, but have a significant foundation where there's something promising, could be there?
Well, I'm not quite sure what venture firm invests in phase 2 trial because it's kind of pretty late in the game. But so I guess I don't really have a clear answer for you. I see the quality of the science, tractability of the problem, of the scientific challenge, and unmet need. And then it doesn't matter what it is. It will, if you execute right, you will build value. So I'm not too worried about stage. We don't do that. I think, look, pharma companies today typically acquire companies before phase 1. So, phase 2, I mean, I'd love to talk offline about the example. I can't imagine an example of someone investing in phase 2, unless it's a public investor who invests in an IPO to go finance phase 2.
Crossover funds, others. So we're out of time, and let me, on behalf of Lynne and myself, thank really a great discussion, Jean-Francois, José, and Ryan. Thank you.